Welcome to Excellent Grades Academy. This is Dr. Byson AM. Today we are doing a 50 biochemistry question marathon. Acknowledgement to Mr. Mwale for giving us these questions. Baby Single, a shout out to you. All right, let's get into this video and see how much biochemistry we have learned. Yes. Okay, so question 1. Which of the following makes water a liquid at room temperature? So which of the following makes water a liquid at room temperature? Non-covalent bonds, hydrogen bonds between water molecules. Correct. So this is the answer here. Hydrogen bonds between water molecules will make water a liquid at room temperature. Van der Waals interactions, there are no Van der Waals interactions. Covalent bonds are intramolecular bonds and they are not intermolecular bonds. Okay, let's move on. The heat of vaporization of water is 540 kgs per gram, 580 kgs per gram, 1060 joules per gram, 600 kilojoules per gram. So the answer is this one here, 540 kilojoules per gram. So when this is the heat that is required for 1 gram of water to turn into vapor. You know that vaporization is simply turning liquid water to vapor. So to vapor, you know, vapor is simply gas. So now per 1 gram of water you require 540 kilojoules of heat. So that's why we say water has got a high heat of vaporization. Just look at how much energy goes into turning 1 gram of water into vapor. Okay. Number 3. The degree of ionization does not depend on which of the following parameter? 1. Nature of solvent. 2. Temperature. C. Concentration. D. Current. So the answer here is current. For ionization to take place, it doesn't depend on current, but it depends on nature of the solvent, the temperature, and the concentration. Number 4. Which of the following mixture in aqueous solution of equimolar concentration acts as a buffer? So this is excess ammonium hydroxide and hydrochloric acid, and then you have hydrogen nitrite and sodium hydroxide. Then you have methanoic acid here plus sodium hydroxide excess, and you have sulfuric acid and potassium hydroxide. So the answer here is ammonium hydroxide excess plus hydrochloric acid. So what is going to happen is that if these react, what you're going to have is ammonium chloride plus water. So in your solution, you're going to have ammonium chloride and ammonium hydroxide, because ammonium hydroxide here was in excess. So it is going to be in solution like this. So this is going to be the equimolar concentration of a solution. Okay. 5. Based on which of the following enzymes hydrolysis reactions are catalyzed? Hydrolysis are catalyzed by hydrolase enzyme. There are no two ways about it. Which of the following is an amino acid? So these are amino acids, and the one that has an amino group is fluorine. Is fluorine. So fluorine is considered as a secondary amino acid, and it has this group here that is attached to it. So it has this group as a side chain that is attached to it. Then you have NH and then OH. This is a secondary amino acid. So secondary amino acid. All right. Number 7. Which of the following is a 39 residue hormone of the anterior pituitary gland? The anterior pituitary gland, which is called the adenohypophysis, produces a lot of hormones. Glucagon is not a 39 residue hormone, but corticotropin is. Okay. Corticotropin is. Corticotropin is a hormone that will go and stimulate the adrenal gland. Number 8. Which of the following statements is correct about column chromatography? A. Resolution improves as column length increases. Mobile phase is a porous solid material kept in the column with appropriate chemical characteristics. No. Stationary phase is a buffered solution that percolates through the mobile phase. No. Large proteins exit the column faster than tiny proteins. So the answer is here. Resolution improves as the column length increases. So as the column length increases, what you start seeing is that the ink will be widening and the colors will be coming in shape. Number 9. Who deduced the double helical structure of DNA? This is a first year question. It is Watson and Francis Crick. Number 10. The amino acid sequences of thousands of different proteins from many species have been determined using principles derived by Sanger. So the full name of this scientist is called Frederick Sanger. Frederick Sanger. This is the guy who gave us ideas on how to determine amino acid sequences in proteins. Okay. The stability of an alpha helix is not affected by which of the following? Bulkiness. B. Occurrence of alanine and glucose and glycine molecules. So this is the answer here. The occurrence of alanine and glycine molecules do not affect the alpha helix, but the occurrence of purine. So if there is purine and there is glycine, then the structure of the alpha helix is affected. In fact, when you reach purine, that's when the alpha helix terminates. Number 12. Which of the following is abundantly found in collagen? It's glycine. So glycine is the amino acid that is found abundantly in collagen. Number 13. Which of the following is a function of a chaperone protein? So a chaperone protein, it rescues proteins that have been folded improperly and allows them to fold properly. So the answer is D here. So that is the function of the chaperone protein. Number 14 is the relationship between oxygen concentration and the fraction of binding sites occupied in myoglobin can be described as, so it can be described as hyperbolic. So when you look at that equation and you plot, you find that it is a hyperbolic curve. Okay. 15. When compared to the first immune response to the same antigen, which of the following is not a hallmark of the secondary immune response? So antibody is generated first. This is true. Many antibodies are produced. This is true. Antibody is generated without T helper cells. Antibody produced has a greater affinity for the antigen. So the answer here is this. So antibody is generated with the helper of the T helper of the T helper cells. All right, 16. When the velocity of enzyme activity is plotted against substrate concentration, which of the following is obtained? It is a hyperbolic curve. So a hyperbolic curve is obtained when the velocity of an enzyme activity is plotted against substrate concentration. Okay, so you should go through these tables. They are very important. Which of the following catalyzes the reversible degradation of 2-phosphoglycerate to phosphoenolate pyruvate? So enolase is the answer here. You're going to see this when you study carbohydrate metabolism under glycolysis. Okay. You do this under glycolysis. Which of the following is an example of epimers? Mannose and glucose, glucose and ribose, galactose and mannose, glucose and galactose. So epimers here, the answer is glucose and galactose. We know that galactose is an epimer of glucose at carbon number 4, C4. But mannose is also an epimer of glucose at carbon number 2. Okay, so here, even this answer makes sense, is correct. Okay, so there were two answers here. This is an epimer at carbon number 2. So mannose is an epimer of glucose at carbon number 2. Whilst galactose is an epimer of glucose at carbon number 4. Number 19. In which of the following glucose residues are linked by beta 1,4 glycosidic bonds? Starch contains alpha glycosidic bonds. Cellulose is the one that contains beta 1,4 glycosidic bonds. Number 20. Which of the following are important for white blood cell recognitions? So they are glycoproteins. So glycoproteins are the ones that act or serve as receptors. So whenever you see receptors on the surface of the cells, those are glycoproteins. Number 21. Which of the following is true about phosphodiester bonds? So we know that phosphodiester bonds, the phosphate group on carbon number 5 goes and joins with the hydroxide group at carbon number 3. So which is this one here? 5-phosphate group of one nucleotide unit is joined to 3' hydroxyl group of the next nucleotide. So the answer here is this. 22. Arrangement of nucleotides in DNA can be seen using which of the following instruments? So we use an X-ray crystallography. That's what we use. 23. Which of the following involves carrying genetic information from DNA to protein synthesis? It is mRNA. Which of the following is true about Tm? So Tm simply means the melting point. So the melting temperature. The melting temperature. So the melting temperature is increased by the higher content of the G2 cytosine base pairs. Okay. So the higher the content of the G to C base pairs, the higher the melting temperature. Why? Because this pair, base pairing, consists of 3 hydrogen bonds. So because here there are 3 hydrogen bonds. Which of the following is true about the Z-DNA helix? It has fewer base pairs per turn than B DNA. This is correct. So it tends to be found at a 3'. No. It has alternating GC sequences. No. It is a permanent conformation of DNA. So the answer here is A. Which of the following involves carrying genetic information from DNA for protein synthesis? So this is a similar question that we have done. The answer here is mRNA. 27. Which of the following enzymes is used in PCR? We use taq-DNA polymerase. taq-DNA polymerase is obtained from Thymus aquaticus. Okay. Aquaticus. This protein is obtained from this species. 28. The recombinant DNA construction involves cleaving DNA with a restriction, in the nucleus and joining with ligase. So this is what recombinant DNA construction involves. Which of the following is termed as conserved DNA order? It is called synteny. So all of this, microarray, orthology, paralog, involves DNA changing the order. But in synteny, you conserve the order of DNA. The number of milligrams of potassium hydroxide required to neutralize the free and combined fatty acid in 1 gram of a given fatty acid is what we call saponification number. So this is called saponification number. Which of the following membrane lipids have a direct glycosidic linkage between the head group sugar and the backbone glycerol? So we have a sugar, which is a glyco, and then we have a connection to glycerol, which is a lipid. So it's a glycolipid. So the answer here is glycolipid. Which of the following membranes contain low cholesterol and high cardiolipin? So it is the inner mitochondrial membrane of a hepatocyte. Inner mitochondrial membrane of the hepatocyte contains low cholesterol and high cardiolipin. Which of the following is a choline containing lipid? It is sphingomyelin. The answer is sphingomyelin. Which of the following proteins does not function in cell-to-cell interaction? So this one does, cytochrome C doesn't. Integrin does, NCAM does. So the answer is cytochrome C. Which of the following is the study of energy relationships and conventions in biological systems? This is called bioenergetics. So the definition of bioenergetics is simply the study of energy relationships and conversions in biological systems. 36. Anabolism and catabolism are chemically linked in the form of ASP, ADP, ATP, phosphodiester linkage. The answer here is ATP. Anabolism involves, so this involves ATP intake. So it takes energy. Whilst catabolism involves ATP output. So ATP output, that is catabolism. Which of the following is not a flavoprotein? dehydrogenase-Qreductase, B. succinate dehydrogenase, xanthine oxidase, cytochrome C. So the answer is cytochrome C. So a flavoprotein involves uptake of involves participation in oxidation reactions in which the electrons are lost. But then when you look at the function of cytochrome C, it actually receives the electrons. That's why it's not a flavoprotein. 38. Which of the following is not a factor determining the activity of an enzyme? Nucleotides are not there. Association with the regulatory protein, yes. Sequestration, yes. Allosteric reaction, yes. So these three are involved, but not nucleotides. 39. Which of the following is a measure of the effect of an enzyme's concentration on flux through a multi-enzyme pathway? Metabolic control, no. Response coefficient, no. Elasticity coefficients, no. But flux control coefficient. So this is the measure of the effect of an enzyme's concentration on flux through multiple enzyme pathways. Okay. Which of the following mixture in aqua solution of equimolar concentration acts as a buffer solution? So we already looked at this question. The answer is A here. 41. Enthalpy refers to the heat released or absorbed during a reaction. Correct. So this is enthalpy. The randomness of or disorder of a system, this is entropy. This is what we call entropy. The overall energy content of a molecule, that gives free energy. The stability of a reaction. In a coupled reaction, the overall spontaneity of a reaction is determined by, so it's determined by the change in Gibbs free energy values of individual reactions. This is what determines the overall spontaneity of the reactions. Okay. Entropy is a measure of the heat released or absorbed during a reaction. No, that's enthalpy. Entropy is the randomness or disorder of a system. That's what we call entropy. So when we say you are full of entropy, we're saying that you are disorderly. Enthalpy refers to, okay, we looked at this already, which is the heat released or absorbed during a reaction. Number 45. The standard free energy of a reaction is a measure of the rate of a reaction, the equilibrium constant of the reaction, the overall energy released or absorbed during the reaction, the activation energy. So this is the overall energy released or absorbed during the reaction. 46. Oxidation reduction, which are redox reactions, involve the transfer of electrons. Definitely. Not protons, not oxygen atoms, and not hydrogen atoms. 47. Energy-rich compounds such as ATP store energy in ATP is simply adenosine triphosphate. So when you look at this, there are three phosphates. There are three phosphates atoms. So the energy is stored in phosphate bonds. So the energy is stored in phosphate bonds. NAD, NADP, and FAD are examples of what we call coenzymes. So these are coenzymes. Cofactors are metal ions. Metal ions. E.g., you've got calcium, magnesium, zinc. So these are the cofactors. Substrates are just molecules that enzymes act on. 49. In bioenergetics, the term chemiosmosis refers to the movement of chemical substances across the cell membrane. No. The generation of ATP using the energy from an electrochemical gradient. So this is the answer here. This is what chemiosmosis is. The generation of ATP using the energy from an electrochemical gradient. The last question in this marathon of questions in biochemistry is the conversion of NAD plus to NADH involves the transfer of, an electron. All right, so this was the 50 question biochemistry marathon in preparation for your assessment. The other 50 question biochemistry marathon will be in the next video. Okay, so the next segment of the questions will be the next video. You can register with us for lessons on 0975 497790. This is Dr. Byson EM, bringing you nothing but the best. Let's medicine together. See you in the next video.